All right. Thank you so much for having us. And yeah, I apologize. Stephanie was unable to make it at the last minute, but hopefully I can try to fill in her shoes. That's and tell you a little bit about what we're up to and yeah, trying to do with Cell Ag for something other than meat, I guess. So a little bit about Jellatech, for those who don't know, we're a pretty small startup still. We were founded in 2020 and we're based here in Raleigh, North Carolina, which is part of the greater sort of research triangle. And as as we were just talking about a second ago, it's been very exciting to see the number of startups and interest from universities and everything in this area sort of explode in the last year since I've been involved in it. I am a biomedical engineer by training, but joined Jellatech about a year ago and or a little over a year ago. And yeah, it has been very exciting to be part of this field. We are sort of a team of scientists and entrepreneurs who have sort of expertise in the areas listed here. And as you probably gathered from the title, we are interested in using Cell Ag to produce the protein collagen, among other things that we're also working on. But our first sort of product is collagen. So you're probably familiar with some of the application of collagen. It is sort of an interesting protein in that it sort of, and this is kind of what I'm going to talk about today, spans some of the, spans several different worlds. Some of them include, the food and beverage sort of areas where collagen is often used, for example, as gelatin to give protein or to give foods sort of structure, texture. It's also, used in the sort of nutraceutical and cosmetics industries. And then in addition to that, it's also has a wide range of uses in the biomedical side of things. And we are interested in sort of producing collagen that will sort of span all of these. And we're doing that. And well, I should say that so many of these applications require that the collagen that we produce, and I'm going to talk a little bit more about the protein in a second, but that it is a fully functional protein. I'll explain a little bit more what that means, but that includes not only on the biomed side, but also on the food and beverage side. For example, to create gelatin, you need a protein that will denature in the right way to provide the sort of strength and texture that's needed for that application. So we're trying to use Cell Ag to generate collagen that can meet these needs. And the gelatin and collagen market is growing rapidly across all those fields, sort of applications listed. And traditional agriculture has been used sort of to meet that demand to this point, primarily. And however, as people in this audience are probably very familiar, it's going to be very difficult to sustain and meet that demand, even the existing demand, let alone the growing demand. And we hear this from a lot of people, customers, potential customers who are interested in our product, saying we don't foresee being able to meet the demand in only a short amount of time going forward. So we've taken the approach of trying to produce collagen with cells in order to meet that demand, and in addition, of course, reduce a number of sort of the bad things associated with traditional agriculture, including climate cost, consistency, which is also a very big thing, particularly, well, depending on the application, and then also sort of from an animal welfare standpoint. I just want to talk quickly about a quick overview of collagen, just because sort of to highlight why we're taking the approach we are and why we think it's beneficial. And so collagen, as you probably know, is a triple helical protein that's actually fairly complex. And it's made of the 3 different alpha chains, which are often the product of multiple genes, actually. And then these chains need to come together inside the cell, be post-translationally modified, be chaperoned so they meet each other, fold, be modified so they're stabilized, cleaved as they exit the cell. And this is sort of a pretty complex process that's been hard to replicate in sort of other systems. And we've taken the approach of using mammalian cells because they have all the machinery to make the protein. And it turns out actually make it fairly efficiently. As you know, the human body is, I think, approximately 30 or 40% collagen by a dry weight. And so in order to make that amount of protein, your cells need to be fairly proficient at it. So that is our approach, and why we've taken the approach of using mammalian cells to produce what we have sort of termed bioidentical, and that it's structurally and functionally the same as native collagen that's found in animal tissues. We can do it then with a high purity. We can have consistent, repeatable results, and we can also, as I'll talk about in a minute, tailor it to specific species. So we began our process, or sort of our journey, with producing bovine collagen. And as I'll talk about in a moment, we've expanded that to human collagen now. Our process is sort of what I guess might be sort of traditional cell ag process, you might think of, in that we remove a small number of cells from an animal and go through a selection and sort of process that allows us to capture the best collagen producers and treat them in a certain way so that they maximize their collagen output, and culture them at high densities in a bioreactor for a short amount of time, where they produce a large amount of collagen that we can then separate from the cells and purify, go through a series of purification steps where we end up with a collagen product at the end. And sort of the human side of this is, it's a little weird to say human and cell ag sort of, or agriculture in the same way, but collagen sort of spans these two worlds a little bit, in that it's can be both used in the food and beverage side of things and the biomedical side of things. And that actually, we think, has some advantages for us as we're sort of developing and scaling up our process. On the food and beverage side, things are typically animal derived, and has typically bovine or porcine, whereas on the biomedical side, it's advantageous to use human collagen for a lot of applications, although people do use some animal collagen. From our perspective, then, we have been able to sort of, as we've been sort of figuring out our process and things like that, benefit from all the economies and scale developments that have been happening in the cell ag industry, which even in my short time in the field have sort of been accelerating like crazy. But then we still are able to, since our collagen is fully functional, try to meet biomedical sort of demands, which give us more of a high price point, and we can and need smaller quantities. So that allows us a little bit of a way to sort of scale up at a slower a little bit of a slower pace. And importantly, human collagen sources are quite limited. So there's a definite need for sort of other sources of human collagen. And then as we scale up further, hopefully we can then transition to also meet the food and beverage side of things as well. And like I said, this sort of a little bit covered before, but with human collagen, then we can have a readily available product that we can produce on demand. It will have much lower variability, we believe. We can keep contaminants out of it that would normally potentially get introduced outside of, say, a lab environment. And then the allogeneity, the human, the fact that it's a human protein being used in a human body would, we think, reduce the risk of sort of unwanted immune responses. Some of the applications include anything from dermal fillers, implant coatings, tissue engineering, and regenerative medicine, collagen scaffolds and sponges for wound healing, injections for arthritis repair, scaffolds for tissue engineering, and 3D sort of bioprinting of scaffolds. And we're, interested in and believe that our collagen should be able to be applied to any of those areas. So with that, we have basically taken our same sort of platform technology that we've used to generate our bovine collagen and extended it now to human collagen we have a cell line now that is a excellent collagen producer. And we have essentially carried over this same process from bovine to human. And I would say I wanted to point out a few, I guess, key differences maybe with this sort of approach relative to, say, the alt protein or sort of meat areas of cell ag that might be maybe, or that we've come ran into and sort of had to strategize for. Human collagen, like I mentioned earlier, is smaller quantities are needed, higher price points are available, and so hopefully we can generate actually some revenue, hopefully at the pilot scale, which we're working towards. Our customers probably in this market are biomedical corporations that are using either other sources of collagen that are very interested in having a sort of cell-derived consistent supply. And so this is not, say, compared to cultured meat where people are sort of marketing directly to the consumer. We're dealing more now in sort of corporate partnerships in this area and working out ways to supply biomed corporations with the product. And this will likely require clinical trials, not paid for by us, but working with these companies to actually evaluate it in a clinical setting. So far to date, then, just kind of to give you a summary of sort of where we stand as we're progressing. We have now, developed cell lines for both bovine and human collagen production. We have continued to sort of optimize our production levels, and that's a ongoing and never-ending process. And then we're able to, extract, purify, process it. We verified in a number of ways that our collagen is triple helical and has a lot of the same function as native collagen. And we've been, for probably the past 6 months or so, supplying various people with samples for evaluation and getting some great feedback from that. And looking to this, what comes next for us, we're basically interested in working to transition what we're doing now to the pilot scale, which is a significant challenge, and also working to test and basically continue to show the functionality of what we're making, specifically now that we're making human collagen for biomedical applications. Yeah, I think that is hopefully about the right amount of time. And I just wanted to say, we have a great, really great team who have done all the work to make some great progress in a what I feel is a pretty short amount of time. Yeah, it's been fun to be a part of it. And I'd be happy to take any questions.